There is a forest in British Columbia where a single tree is simultaneously feeding 14 of its neighbors, sending chemical warnings to seedlings it has never touched, and flooding the root systems of dying competitors with enough sugar to keep them alive through the winter. None of this is visible. None of it makes a sound. It happens entirely underground, in a web of living threads so fine that a single teaspoon of forest soil can contain more than 7 miles of them, threaded through a darkness where no light has ever reached. We have spent centuries looking at forests and seeing individuals, tall trees, short trees, old trees, young trees. We built entire sciences around counting them, measuring them, naming them by species and categorizing them by growth rate. We mapped their canopies and sampled their soils. We debated their timber value and catalogued their medicinal properties. And through all of it, we missed what was happening right beneath our feet. Because trees are not individuals, they are nodes. They are connection points in a network so ancient, so vast, and so functionally sophisticated that the most accurate comparison available to human language is not a forest, it is a brain. The mycorrhizal network, the underground web of fungal threads that links the root systems of trees across entire forest ecosystems, transfers carbon, nitrogen, water and chemical signals between organisms that have no nervous system, no brain, and no apparent mechanism for communication. And yet the communication happens, the transfers happen, the responses happen, with a precision and a coordination that has forced biologists to fundamentally reconsider what a forest is, what an individual organism is, and what the word intelligence means when it is stripped of every assumption we have inherited from studying animals. If this challenges what you thought you knew about the natural world, if it makes the ground beneath your next walk through any patch of trees feel suddenly alive in a way it did not before, then consider tapping that like and subscribe button. It helps more than you know, and I genuinely appreciate every single one of you. Now, get yourself comfortable. Let's begin. Chapter 1. The discovery that changed everything. In 1997, a forest ecologist named Suzanne Simard published a paper in the journal Nature that the scientific establishment was not entirely prepared to receive. Simard had spent years working in the forests of British Columbia, first as a reforestation scientist for the Canadian logging industry, and later as a doctoral researcher at Oregon State University. Her work had begun with a practical industrial question. The timber industry had a problem. When old-growth forest was clear-cut and replanted with commercial Douglas fir seedlings, those seedlings consistently underperformed. They grew more slowly than expected, showed higher mortality rates, and seemed to struggle even when soil conditions appeared adequate. The standard forestry solution was to remove the competing vegetation, particularly birch trees, which were considered weeds in a commercial Douglas fir plantation. Simard suspected this was wrong. She suspected, based on years of observation and an accumulating body of laboratory research into soil fungi, that the birch trees were not competitors at all. She suspected they were partners. To test this idea, she designed an experiment that would become one of the most cited and most controversial studies in modern ecology. She planted seedlings of 3 species, Douglas fir, paper birch and western red cedar, in forest plots in the interior of British Columbia. She then injected individual trees with radioactive carbon isotopes and stable carbon isotopes, different tracers for different trees, and waited. The results were unambiguous and, to many of her colleagues, shocking. Carbon was moving between trees. Specifically, carbon injected into birch trees was appearing in the root systems and foliage of nearby Douglas fir seedlings. Carbon from the fir was moving in the opposite direction into the birch. The cedars, which had a different type of fungal association, showed no such exchange, but the directionality of the transfer was not random. It responded to conditions. When Simard shaded the Douglas fir seedlings, reducing their photosynthetic output and their ability to fix their own carbon, the transfer from birch to fir increased. When the birch was shaded instead, the direction reversed. The trees were not simply leaking carbon into a shared pool, they were sending it, and the amount sent responded to which tree needed it more. The paper was titled Net Transfer of Carbon Between Ectomycorrhizal Tree Species in the Field. It was published in Nature's August 7th issue, and it immediately generated a scientific argument that has not fully resolved in the nearly 3 decades since. Some ecologists embraced the findings as confirmation of what field observation had been suggesting for years. Others pushed back hard, arguing that Simard's methodology could not distinguish between active transfer through fungal networks and passive diffusion through soil water, or between mutually beneficial exchange and simple parasitism by fungi extracting carbon from both trees simultaneously. The debate was productive. It drove a generation of follow-up research using increasingly sophisticated methods. Genetic analysis of fungal communities, stable isotope tracing at higher resolution, manipulation of specific fungal networks through targeted disruption, controlled greenhouse experiments that isolated individual variables. And as the methods improved, the picture that emerged was more complicated than Simard's original study, more nuanced than the popular press coverage that followed, and more extraordinary than either side of the original debate had anticipated. The forests were talking. The question was what exactly they were saying, and how. Chapter 2. The Architects of the Network. To understand how trees communicate, you first need to understand what the mycorrhizal network actually is, because the word network, borrowed from human technology, imports a set of assumptions that do not all apply here. A mycorrhiza, the word comes from the Greek for fungus and root, is a symbiotic association between a fungus and the root system of a plant. These associations are ancient. Fossil evidence places mycorrhizal relationships at approximately 450 million years ago, around the time the first land plants were colonizing the terrestrial surface. Current genomic analysis suggests the relationship may be even older. The oldest known land plant lineages all form mycorrhizal associations, which has led many researchers to conclude that plants did not colonize land alone. They did it with fungal partners. The partnership is as old as life on land itself. There are several types of mycorrhizal associations, but 2 dominate the forest ecology literature. Ectomycorrhizal fungi, which are common in boreal and temperate forests, wrap a dense sheath of fungal tissue around the outside of fine root tips without penetrating the root cells. Arbuscular mycorrhizal fungi, which are more common in tropical forests and grasslands, actually enter the root cells and form branching structures called arbuscules inside them. Both types extend networks of fungal threads called hyphae out into the surrounding soil. These hyphae are extraordinary structures. They are typically between 2 and 10 micrometers in diameter, far finer than a human hair, which averages around 70 micrometers. A single fungal individual can extend its hyphal network across several square meters of soil or, in the case of some large fungal organisms, across hectares. The famous Armillaria ostoyi specimen in the Malheur National Forest in Oregon occupies an estimated 2,385 acres and is estimated to be between 2,000 and 8,000 years old. It is the largest known organism on Earth by area. But Armillaria is a pathogen. The symbiotic mycorrhizal networks in a healthy forest are not single organisms, they are communities. A cubic meter of forest soil may contain thousands of meters of hyphae from dozens of fungal species, all interwoven, many of them connected to multiple tree species simultaneously, forming a multi-species, multi-individual network with no central controller, no headquarters, and no single point of failure. The hyphae extend the effective absorptive surface of tree roots by factors of 100 to 1,000. A Douglas fir seedling with mycorrhizal associations accesses soil nutrients across a volume of soil many times larger than its root system alone could reach. The fungus, in exchange, receives carbon, primarily in the form of sugars, from the tree's photosynthesis. This is the basic exchange that was well understood long before Simard's experiment. The tree feeds the fungus. The fungus feeds the tree. A simple, well-documented mutualism. What was not well understood was what happened when the hyphae of fungi associated with one tree connected through the shared fungal network with the hyphae of fungi associated with a different tree. What happened at those junctions, which researchers began calling common mycorrhizal networks, or CMNs, turned out to be the answer to a question nobody had thought to ask, precisely because nobody had suspected the question was worth asking. What happens when trees touch? Chapter 3. The chemistry of an underground conversation. Carbon transfer between trees through mycorrhizal networks is now supported by dozens of independent studies using multiple methodologies. The debate has shifted from whether it happens to what governs it, how significant it is relative to other carbon pathways, and what functional role it plays in forest ecology. But carbon is only one of the molecules moving through these networks, and in many respects, it is the least interesting one. Nitrogen is the element that most constrains plant growth in many terrestrial ecosystems. It is abundant in the atmosphere. Roughly 78% of the air you are breathing right now is nitrogen. But atmospheric nitrogen is in a form that plants cannot use directly. The conversion of atmospheric nitrogen into biologically available forms, a process called nitrogen fixation, requires either specialist microbes or the slow decomposition of organic matter. Forest soils hold enormous reserves of nitrogen locked in organic compounds, and mycorrhizal fungi are among the primary agents that mobilize this nitrogen and make it available to trees. Studies in boreal forests have documented nitrogen moving through mycorrhizal networks from decomposing organic material and from nitrogen-fixing bacteria associated with certain tree species into the root systems of trees that cannot fix nitrogen themselves. The fungal network acts as a distribution infrastructure, moving limiting nutrients from areas of relative surplus to areas of relative deficit. Water is transferred through mycorrhizal networks as well, though this pathway is less well characterized. Hydraulic redistribution, the movement of water from moist soil layers to dry ones through root systems and associated fungi, has been documented in multiple forest systems. During drought conditions, deep-rooted trees can transfer water upward through their root systems and outward through mycorrhizal connections to shallower-rooted neighbors. The network functions, in these conditions, as an irrigation system operated by the forest community for the collective benefit of its members. Phosphorus, potassium, calcium, and various micronutrients also move through mycorrhizal networks, though the magnitudes of these fluxes and their ecological significance vary enormously between forest types, tree species, and fungal communities. But what genuinely upended the scientific understanding of forest ecology was not the transfer of nutrients. Nutrients moving through soil is, at some level, expected. Soil is a medium. Things diffuse through it, get carried by water, get mobilized by biological activity. What shocked the field was the discovery that the chemical signals traveling through mycorrhizal networks appeared to carry information. Not carbon, not nitrogen, not water. Information. The signal that first alerted researchers to this possibility came from studies of plant defense responses. When plants are attacked by herbivores or pathogens, they initiate chemical defense responses. They produce compounds toxic to the attacker. They strengthen cell walls. They call in predators of the attacker by releasing volatile chemicals that attract parasitoid wasps or predatory insects. These responses are well documented in above-ground plant communities and involve the release of volatile organic compounds into the air that neighboring plants can detect and respond to by preemptively activating their own defenses. This above-ground chemical communication was already remarkable enough, but researchers studying the soil connections between plants began to ask whether a similar signal might travel through mycorrhizal networks, and the answer turned out to be yes. Chapter 4. Warnings through the dark. In 2010, a research team led by David Johnson at the University of Aberdeen published a study demonstrating that broad bean plants connected by mycorrhizal networks showed enhanced aphid resistance even when the connected plants had no direct exposure to aphids. An infested plant was transferring a chemical defense signal through the shared fungal network to its connected neighbors. Those neighbors responded by activating defense mechanisms, specifically by producing more of the volatile compounds that repel aphids and attract aphid predators before any aphid had touched them. The signal traveling through the network was not a nutrient. It was not a building block. It was a warning. Subsequent studies expanded on this finding across multiple plant species and multiple types of stress. Plants connected by mycorrhizal networks have been shown to transfer signals that prime defensive responses against fungal pathogens, against drought stress, and against herbivory by caterpillars, beetles, and other invertebrates. The signal transfer is faster than the spread of the threat itself, giving connected plants time to prepare before attack arrives. The mechanisms underlying these signal transfers are not fully elucidated. Several pathways have been proposed and have supporting evidence to varying degrees. Hormonal signaling molecules, particularly jasmonates and salicylates, the same chemical families involved in above-ground plant defense responses, have been detected at elevated levels in plants connected to attacked neighbors through mycorrhizal networks. Small RNA molecules have been proposed as signal carriers, as have electrical signals propagating along hyphal membranes. The fungal network in this model functions as a signal cable, with chemical information entering at one tree's root system and emerging at another's. The specificity of these signals is a critical and contested question. Critics of the stronger interpretations of mycorrhizal communication point out that demonstrating transfer of a chemical compound through a fungal network is not the same as demonstrating that the compound carries adaptive information. A chemical might move because it follows concentration gradients, not because the sending tree is deliberately warning its neighbors. The receiving tree might respond because the chemical is a generic stress indicator, not because it has decoded a specific message. This is a legitimate scientific objection, and it highlights the danger of using language borrowed from human communication to describe processes that may be fundamentally different in their underlying mechanism. Trees do not have brains. They do not have intentions. They do not, in any sense we currently understand, decide to warn their neighbors. What they do is produce and release chemical compounds whose effects, in the context of evolution, have been shaped by millions of years of selection pressure. The result of that selection pressure, operating at the level of the entire connected network, produces outcomes that look, from the outside, strikingly like communication. Whether we call it communication depends on our definition. Whether we acknowledge its functional reality requires only that we look at the data. Chapter 5. The Mother Tree. Among the most provocative claims to emerge from forest network research is the concept of the mother tree. The hypothesis that the largest, oldest trees in a forest act as hub nodes in the mycorrhizal network, disproportionately connected to more trees, transferring more resources and preferentially directing resources to their own offspring. Suzanne Simard's later research, conducted after she moved to the University of British Columbia, focused extensively on this hypothesis. In a series of experiments involving Douglas fir forests, she documented that the largest, oldest trees, the trees with the most extensive root systems and therefore the most extensive mycorrhizal connections, were linked to a significantly higher number of neighboring trees than younger, smaller individuals. These hub trees, which she termed mother trees, appeared to play a structurally critical role in the network's connectivity. The mother tree hypothesis gained significant popular attention following Simard's 2021 book, Finding the Mother Tree, which presented her research alongside a memoir of her experiences in the forests of British Columbia. The book reached a wide audience and brought the concept of forest communication into mainstream cultural conversation in a way that no academic paper had achieved. The scientific ecology that community's response was more measured. The hub tree observation that large old trees have more mycorrhizal connections than small young ones is reasonably well supported and not particularly surprising. Larger root systems create more opportunities for fungal association, and older trees have had more time to develop extensive networks. What is more contested is the claim that these hub trees preferentially direct resources toward kin, toward their own offspring, and the claim that losing a hub tree causes measurable network disruption that harms the broader forest community. Studies on kin recognition in plants have produced interesting results. Some research suggests that plants can distinguish their own root exudates from those of unrelated individuals of the same species, and that this recognition influences competitive behavior. If true, it would suggest a mechanism by which mycorrhizal resources might be preferentially routed toward genetically related individuals. But the evidence for kin-directed resource transfer through mycorrhizal networks specifically remains disputed, with some studies supporting it and others finding no kin preference in carbon transfer. The hub tree disruption hypothesis, the idea that removing a large old tree degrades the connectivity of the entire local network, has more direct relevance to forestry practice. Several modeling studies and some empirical work suggest that forest network connectivity is sensitive to the loss of highly connected nodes, paralleling findings from network theory in other contexts. If true, this would have direct implications for selective logging practices, that preferentially remove the largest, commercially most valuable trees, precisely the trees most likely to function as hubs in the mycorrhizal network. Whether the empirical evidence currently available is strong enough to support a firm management prescription is a question on which ecologists disagree. What is not in dispute is that the question is worth asking, and that the answer has consequences for how we manage the forests that cover approximately 31% of Earth's total land surface. Chapter 6. The Underground Economy. To understand the full complexity of what mycorrhizal networks do, it helps to think of them not as a telephone system, but as an economy. A living market where resources flow according to supply, demand, and the terms of long-standing biological contracts. The basic currency of this economy is carbon. Plants fix carbon through photosynthesis, converting atmospheric carbon dioxide into sugars using solar energy. Those sugars are the primary energy source for the fungal partners, which cannot photosynthesize. In exchange, the fungi provide mineral nutrients and water that the plant's root system alone cannot efficiently access. This is the foundational transaction, and it has been operating for roughly 450 million years. But the economy is not bilateral. It is not a simple two-party trade between one tree and one fungus. A single tree is typically associated with dozens of fungal species simultaneously. A single fungal individual is typically associated with multiple tree species. The result is a market with many buyers, many sellers, and many goods. A market in which the price of any given commodity is determined by the relative abundance and need of the participants. The implications of this market structure are significant. In a diverse forest ecosystem, trees of different species with different phenologies, different drought tolerances, different light requirements, and different seasonal growth patterns will have varying carbon demands and nutrient needs at any given time. A deciduous tree that has shed its leaves and entered winter dormancy is producing no photosynthate and consuming minimal resources. An evergreen neighbor is continuing to photosynthesize, albeit at reduced winter rates, and may be directing carbon into the network during a period when the deciduous tree cannot reciprocate. Over the course of a year, these asymmetries even out. Both species receive benefits from the network when they need them and contribute when they can. The timing mismatch between different species' needs creates a kind of collective buffer, a reserve capacity that smooths resource availability across seasonal variation. No individual tree would maintain this buffer on its own. The network maintains it collectively, distributing surplus from those who currently have it to those who currently need it. This buffering function has been documented most clearly in studies of mixed-species forests. Pure monocultures of a single tree species, which dominate commercial forestry, show different network dynamics than mixed forests, and a growing body of research suggests that species diversity correlates with network resilience. When a monoculture faces a stress, drought, pest outbreak, pathogen, all trees face the same stress simultaneously, and the network has no source of surplus to draw on. When a mixed forest faces the same stress, some species are more resistant than others, and the network can redistribute resources from the resistant to the susceptible. This is not altruism in the human sense. It is an emergent property of a network whose members have divergent responses to the same environmental conditions. The resilience is a structural consequence of diversity, not a decision made by any individual within the system. But the functional outcome is the same. The mixed forest survives stresses that destroy monocultures. Chapter 7. What fungi get from the deal. Any account of the mycorrhizal network that focuses exclusively on the trees risks missing the most important player in the story. The fungi are not passive infrastructure. They are not pipes through which tree resources flow. They are autonomous organisms with their own evolutionary interests, their own competitive dynamics, and their own capacity to influence the terms of the exchange. Mycorrhizal fungi receive between 10 and 30% of a tree's total photosynthetic output as payment for their services. In a large forest, this amounts to an enormous quantity of carbon, far more than the fungi need simply to maintain their hyphal networks. The excess carbon fuels fungal reproduction, competitive exclusion of other fungal species, and the construction of dense hyphal mats in areas of high nutrient concentration. The fungi are not passive recipients. They actively regulate the exchange. Studies of the molecular mechanisms at the plant-fungus interface have shown that the fungal partner influences plant gene expression, upregulating genes associated with nutrient uptake and downregulating certain defense responses that might otherwise limit fungal access to plant tissue. The plant, in turn, regulates the quantity of carbon it delivers based on the quality of the fungal service, providing less carbon to fungal partners that deliver fewer nutrients. This co-regulation means that the mycorrhizal relationship is not fixed. It is a continuously renegotiated contract, with both parties adjusting their contributions in response to what they receive. A plant in nutrient-poor soil that encounters a highly efficient fungal partner will invest more carbon in that partnership. A plant in nutrient-rich soil may reduce its investment in fungal partnerships, a phenomenon called the mycorrhizal dependency reduction that has been documented in soils with high phosphorus concentrations. The fungi also compete with each other for plant partners. Different fungal species have different specializations. Some are more effective at mobilizing nitrogen, others at mobilizing phosphorus, others at extending into deep soil horizons where different resources are available. A tree root system in a healthy forest may be simultaneously associated with dozens of fungal species occupying different ecological niches, collectively providing access to a broader range of resources than any single fungal partner could offer. This competitive community of fungal partners may be one reason why forest diversity is difficult to fully understand from the tree perspective alone. The trees you see above ground are, in a sense, the visible expression of invisible fungal communities below ground. Different fungal communities support different tree community compositions. When disturbance, logging, fire, soil compaction, chemical contamination disrupts the fungal community, the above-ground tree community is altered even if the trees themselves are not directly harmed. The forest you can see is partly a reflection of the forest you cannot. Chapter 8. Electrical signals and the question of plant intelligence. In 2022, a study published in Science demonstrated that plants transmit electrical signals along their vascular systems in response to damage, light changes, and temperature fluctuations. These electrical signals, which bear a functional resemblance to action potentials in animal nervous systems, propagate from the site of stimulus to distant parts of the plant within seconds to minutes, triggering systemic responses that alter gene expression, chemical production, and even growth patterns. This finding did not come from nowhere. Research into plant electrical signaling has a history stretching back to the 1870s, when the physiologist John Burdon Sanderson documented electrical responses in the Venus flytrap. What is new is the resolution at which these signals can now be studied, the genetic tools available to trace their mechanisms, and the growing body of evidence suggesting that electrical signaling is not a curiosity of carnivorous plants, but a general feature of plant biology. The relevance to forest network communication is this. If individual plants transmit electrical signals internally, and if those plants are physically connected to each other through shared fungal networks, then the possibility exists that electrical signals might propagate from one plant to another through those connections. Some researchers have proposed exactly this, that the mycorrhizal network might serve as a medium for interplant electrical communication, not just chemical communication. The evidence for this is preliminary and contested. Measuring electrical signals at the plant-fungus interface is technically difficult, and distinguishing electrical signals propagating through fungal hyphae from chemical signals that induce electrical responses at the receiving plant's root system is a methodological challenge that has not been fully resolved. A 2020 study led by Andrew Adamatsky at the University of the West of England claimed to detect oscillating electrical potentials in mycorrhizal networks of wood-boide species, though the interpretation of these signals and their significance for intertree communication remains debated. The question of plant intelligence occupies an increasingly prominent and contentious corner of contemporary plant science. A group of researchers who call themselves plant neurobiologists argue that plants display forms of learning, memory, and adaptive behavior that are analogous to, if not identical with, animal intelligence. They point to documented examples of habituation, a form of learning in which organisms reduce their response to stimuli that prove to be harmless, in the sensitive plant Mimosa pudica. They point to studies suggesting that plants can solve maze-like resource distribution problems, integrating information from multiple environmental cues to optimize growth patterns. They point to the existence of signaling molecules in plants that are chemically similar to neurotransmitters in animal brains. Critics of plant neurobiology, including a substantial number of plant scientists who signed an open letter in 2007 and an updated response in subsequent years, argue that this framework inappropriately imports animal concepts into plant biology, risks anthropomorphizing organisms that operate on entirely different principles and may mislead public understanding of what plants actually do. They argue that the behaviors attributed to plant intelligence can be fully explained by biochemical responses to environmental signals without invoking anything resembling neural computation. This debate is ongoing and, in its more nuanced forms, genuinely productive. It is forcing both sides to be more precise about what intelligence means, what communication means, and what constitutes an information processing system. Those clarifications are useful regardless of where the debate ultimately lands. What is not in serious scientific dispute is that plants integrate multiple environmental signals, coordinate responses across their entire body in ways that involve information transfer through chemical and electrical pathways, and that networks of connected plants show coordinated responses that no individual plant could produce alone. Whether you call that intelligence is, at some level, a question about language. Whether you take it seriously is a question about science. Chapter 9. The Forest That Remembers. One of the most striking findings in mycorrhizal network research concerns the long-term dynamics of the network itself. The hyphae of mycorrhizal fungi are short-lived structures, typically lasting days to weeks before being replaced. But the fungal individuals generating those hyphae are not. Many mycorrhizal fungi are perennial organisms whose hyphal networks are continuously regenerated from long-lived individuals that persist in forest soil for decades or longer. This persistence has a consequence. The network has memory. Not memory in the neurological sense, not stored information that can be recalled and interrogated, but structural memory, the encoded history of past associations, past carbon transfers, past chemical exchanges. In the form of fungal community composition, network topology, and the distribution of hyphal biomass across the soil profile. Consider what this means in practical terms. A forest that has been growing on a site for a century has a fungal community shaped by a century of plant-fungus interactions. The species composition of that community reflects which fungal species were best able to establish partnerships with which tree species under the specific soil conditions, microclimate patterns, and disturbance history of that site. The spatial distribution of fungal biomass reflects where carbon has been most reliably available from photosynthesizing plants, and where nutrients have been most reliably accessible for fungal mobilization. When a new tree seedling germinates in this forest, it does not enter an undifferentiated soil environment. It encounters a structured fungal community that carries the accumulated legacy of that forest's history. The seedling's access to mycorrhizal partners, the quality and species composition of those partners, and the degree to which it can access the existing network are all shaped by what happened in that forest before the seedling existed. This has direct implications for forest regeneration after disturbance. Studies of clear-cut forest sites consistently show that soil fungal communities change dramatically after logging, with many of the ectomycorrhizal species associated with mature trees declining or disappearing in the years following harvest. When trees are replanted on these sites, the seedlings encounter a degraded fungal community, one that lacks many of the specialist partners that would have been available in an intact forest. The consequence is measurable. Seedlings planted into sites where the original fungal community has been disrupted typically show slower growth, higher mortality, and reduced resistance to drought and disease compared to seedlings growing in intact forest with access to mature fungal networks. The network's historical knowledge, encoded in its species composition and spatial structure, is an asset that takes decades to rebuild. This finding has become one of the strongest practical arguments for retaining large old trees and diverse tree species in managed forests, not for sentimental reasons, not for biodiversity targets measured in species counts, but for the functional reason that old trees maintain the fungal community that makes the entire network work. Chapter 10. The Dark Side of the Network. Science communication about the mycorrhizal network has a tendency to slide into sentimentality, toward a vision of the forest as a cooperative utopia where trees help each other, share resources equitably, and maintain a harmony that human civilization might learn from. This vision is appealing. It is also incomplete. The mycorrhizal network is not a cooperative network in any simple sense. It is a community of organisms with divergent and sometimes conflicting interests, connected by relationships that are sometimes mutualistic, sometimes parasitic, sometimes both simultaneously depending on conditions. Fungi are not always the generous intermediaries that popular accounts portray them as. Under certain conditions, mycorrhizal fungi shift from mutualism to parasitism, extracting carbon from plant partners without providing proportionate nutrient return. This shift is particularly well documented in high-nutrient soils where plants have access to phosphorus and nitrogen without fungal mediation. When the plant's need for fungal services is low, the plant reduces its carbon investment. The fungus, if it can, continues extracting carbon while reducing its service delivery. The relationship becomes exploitative. Carbon parasitism is also documented between plants sharing mycorrhizal networks. A plant in deep shade may be fixing very little carbon through photosynthesis, yet if connected to a mycorrhizal network that includes well-lit neighbors, it may extract carbon from those neighbors without contributing a fair return. This kind of mycorrhizal parasitism or cheating has been documented in several plant species, particularly in mycoheterotrophs, plants that have lost the ability to photosynthesize entirely and survive exclusively by parasitizing the mycorrhizal networks connected to photosynthetic neighbors. The ghostly Indian pipe plant, Monotropa uniflora, is the most striking example in temperate forests. It is completely devoid of chlorophyll, uniformly white or pinkish, lacking any capacity for photosynthesis. It survives entirely by tapping into the mycorrhizal networks connecting neighboring trees, extracting carbon that ultimately originated in those trees' photosynthesis without contributing anything in return. It is a complete parasite, exploiting the cooperative infrastructure of the forest community for its own survival. The existence of cheaters in the system is not a failure of the network's logic. It is a consequence of it. Any system of exchange creates the opportunity for exploitation. The evolutionary pressures that shaped mycorrhizal relationships have not produced a system immune to parasitism. They have produced a system in which parasitism occurs alongside genuine mutualism in proportions that vary across species, conditions, and time. Competition through the network is also documented. Plants can inhibit competitors by releasing allelochemicals, compounds toxic to other plant species, into the soil. Some research suggests that these inhibitory compounds can travel through mycorrhizal networks, reaching target plants that share fungal connections with the sender. The network that carries warnings about aphids can also carry chemical attacks on competitors. The forest is not a utopia. It is an economy, complex, dynamic, structured by competing interests, and shaped by evolution's relentless pressure to maximize reproductive success regardless of what that requires. The cooperation is real. The competition is equally real. Both operate simultaneously through the same underground infrastructure. Chapter 11. The network under siege. The mycorrhizal network is one of the most abundant and ecologically critical biological systems on Earth. It is also one of the most threatened. The threats are multiple and cumulative, and most of them are invisible because the network itself is invisible. When a forest is logged, the loss of trees is obvious. The loss of the fungal community that supported those trees is not. When soil is compacted by heavy machinery, the damage to surface structures is apparent. The disruption to the hyphal networks woven through the top 20 centimeters of soil, the zone of highest mycorrhizal density, is not. Agricultural intensification is perhaps the most widespread threat to mycorrhizal fungi globally. Modern high-yield agriculture typically involves tilling soil, which physically disrupts fungal networks, applying synthetic fertilizers, which reduce plants' incentive to invest in fungal partnerships, and using fungicides, which directly kill fungal organisms, including beneficial mycorrhizal species. A single application of certain fungicide treatments can reduce mycorrhizal colonization of crop roots by more than 90%. Repeated applications over multiple growing seasons can effectively sterilize the mycorrhizal community in agricultural soils. The consequence is a feedback loop. Crops grown in fungicide-treated, fertilizer-dependent soils become dependent on those inputs. When fertilizer supplies are disrupted by supply chain failures, by price spikes, by the eventual limits of phosphate rock mining, a non-renewable resource that many analysts believe will become critically scarce within this century, those crops have no mycorrhizal network to fall back on. The fungal community that could have buffered them against nutrient stress has been eliminated. Nitrogen deposition from industrial agriculture and fossil fuel combustion is altering mycorrhizal communities in forests far from any farm or factory. Elevated nitrogen availability in soils that evolved under nitrogen-limited conditions shifts the cost-benefit calculation of mycorrhizal associations for trees. When nitrogen is cheap, when it arrives from the atmosphere as pollution, trees invest less in their fungal partnerships. Fungal biomass declines. Network connectivity decreases. The forest becomes, structurally, more like a collection of individuals and less like a network. Several long-term monitoring studies in European forests have documented these changes over decades of increased nitrogen deposition. The species composition of the mycorrhizal community shifts away from the deep soil specialists toward near-surface generalists. Network connectivity metrics decline, and forests that were once highly resilient to drought and disease show increasing vulnerability. Climate change adds another layer of disruption. Mycorrhizal fungi are sensitive to temperature and moisture conditions. As climate zones shift, the fungal communities adapted to current conditions may be unable to migrate as quickly as the trees they serve. A Douglas fir, whose range shifts northward or upslope in response to warming, may find itself in soil whose fungal community is dominated by species adapted to different host trees. The partnership that sustained the tree for centuries may not be waiting for it at the new address. Some of the most dramatic evidence of mycorrhizal network disruption comes from forests affected by invasive pathogens. The chestnut blight, introduced to North America in the early 20th century, eliminated the American chestnut from the eastern deciduous forest within decades. But the chestnut's mycorrhizal partners did not simply adopt other hosts and maintain the network intact. The loss of billions of chestnut trees removed a major carbon source from the network. The fungal species that specialized in chestnut association declined. The network reorganized around surviving tree species, but the reorganization took decades and may never fully recover the ecological function of the original chestnut-dominated forest. The parallel for ash trees currently being lost across North America and Europe to the emerald ash borer and ash dieback disease is in progress now. The parallel for white bark pine, declining rapidly due to mountain pine beetle and white pine blister rust under warming climate conditions, is in progress now. Each species loss is also a network loss, a reduction in the diversity of carbon sources and chemical signals flowing through the underground infrastructure of the forest. Chapter 12. The science that surprised its own authors. It is worth pausing to acknowledge what an unusual position the mycorrhizal network has occupied in the scientific literature. Research in this area has surprised its authors repeatedly, not in the way that science usually surprises, by revealing that the effect is larger or smaller than expected, but in a more fundamental way. It has surprised researchers by revealing that the system exists in a form substantially more complex than the conceptual framework they brought to studying it. Suzanne Simard began with a question about carbon transfer between two tree species in commercial plantations. She ended up writing about forests as superorganisms with emergent cognitive properties. This is not the trajectory of someone who set out to confirm a predetermined conclusion. It is the trajectory of a scientist following data into territory for which her training had not fully prepared her. Merlin Sheldrake, the mycologist and author of Entangled Life, who spent years studying mycorrhizal networks in Panamanian tropical forests, describes the experience of encountering fungi in the field as a repeated confrontation with the inadequacy of the conceptual tools he arrived with. The categories organism, individual, network, communication kept breaking down against the biological reality he was observing. The fungi were simultaneously individuals and networks, simultaneously cooperative and competitive, simultaneously suppliers and parasites depending on conditions. This experience is common in mycorrhizal research. The systems are complex in a way that makes straightforward experimental interpretation difficult. Variables that seem controlled turn out to be entangled. Effects that seem robust in one forest type disappear in another. The same fungal species behaves differently in association with different host trees, in different soil conditions, in different seasons. The field has accumulated a substantial body of results that are real but difficult to synthesize into a simple unified theory of how mycorrhizal networks work. This complexity is not a weakness of the science, it is a property of the system being studied, and it has implications for how we communicate this research to a public that is hungry for the story, but often encounters it through accounts that smooth over the complexity in favor of a cleaner narrative. The cleaner narrative: trees talk to each other underground, old trees nurture their children, forests are cooperative superorganisms, is not false, but it is incomplete. It omits the competition, the parasitism, the species-specific variation, the contested findings, and the genuine uncertainty about mechanisms that characterizes the actual scientific literature. This does not make the story less interesting. In many ways, it makes it more interesting. A forest that is both cooperative and competitive, both altruistic and exploitative, both ancient and continuously adapting. A forest that is, in other words, as complex as any other living system we have ever examined in depth, is more fascinating than a forest that is simply kind. Chapter 13. Practical Applications and Future Directions. The research on mycorrhizal networks is not only scientifically significant, it has direct practical implications for agriculture, forestry, ecological restoration, and carbon sequestration. Implications that are beginning to influence policy and practice in ways that would have seemed speculative a generation ago. In agriculture, the understanding of mycorrhizal function has prompted renewed interest in farming practices that preserve or restore soil fungal communities. Reduced tillage and no-till agriculture, cover cropping, the reduction or elimination of synthetic phosphorus fertilizers, and the avoidance of broad-spectrum fungicides are all practices that, among other benefits, support mycorrhizal colonization of crop roots. Field experiments. Comparing high-input conventional agriculture with lower-input systems that support mycorrhizal function have produced mixed results. Yields are often lower in mycorrhizal supportive systems, at least initially, but long-term studies suggest that the yield gap narrows as the fungal community recovers, and that mycorrhizal-supported crops show greater resilience under drought and pest pressure. Commercial mycorrhizal inoculants, products containing spores of mycorrhizal fungi that can be applied to seeds or transplants have become a significant agricultural market. The effectiveness of these products varies considerably depending on the resident soil fungal community, the soil conditions, and the match between the inoculant species and the crop species. In soils where mycorrhizal communities have been severely depleted by intensive agriculture, inoculation can produce measurable benefits. In soils with intact native fungal communities, adding commercial inoculants may produce little effect. The market has raced ahead of the science in some respects, with products making claims that exceed the supporting evidence, but the underlying principle that restoring mycorrhizal function in degraded soils improves plant performance is sound. In forestry, mycorrhizal research has begun to influence practices in commercial timber production, particularly in the Pacific Northwest and British Columbia, where much of the foundational research was conducted. The concept of nurse trees, trees retained during harvest specifically because of their role in maintaining the fungal network for regenerating seedlings, has gained traction in some forestry guidelines. The practice of retaining structural diversity, including large old trees and dead standing timber, is supported by mycorrhizal research as well as by habitat considerations for vertebrate species. Ecological restoration projects, particularly in severely degraded sites such as mining-impacted areas and construction sites, have incorporated mycorrhizal inoculation into restoration protocols with documented success. Establishing mycorrhizal networks early in the restoration process significantly improves the establishment of target plant species because the fungi can be re-establishing before the plant community has fully recovered. Carbon sequestration is perhaps the most potentially significant practical application of mycorrhizal network research. The soil carbon stored in forests, including the enormous quantity of carbon in fungal biomass and in organic matter stabilized by fungal activity, is a major component of the terrestrial carbon budget. Estimates suggest that mycorrhizal networks globally store several billion tons of carbon per year in stable soil organic matter. This is not a trivial contribution to the carbon cycle at a time when global atmospheric carbon dioxide concentrations are rising rapidly. Research into the carbon storage function of different fungal communities is revealing important differences. Ectomycorrhizal networks, which dominate in boreal and temperate forests, appear to promote the formation of stable soil organic matter more effectively than arbuscular mycorrhizal networks, which dominate in tropical forests. This difference may partly explain why boreal and temperate forests, despite having lower overall biomass than tropical forests, store disproportionately large amounts of carbon in their soils. Understanding these mechanisms at the network level, not just the individual plant or soil chemistry level, but the whole network level, may be critical for predicting how forest carbon storage will respond to climate change, disturbance, and management intervention. The models we currently use to project forest carbon futures are, with few exceptions, models that do not include mycorrhizal network dynamics. They treat trees as independent organisms, drawing nutrients from a uniform soil matrix. The real forest does not look like that, and our carbon projections may be systematically wrong because of this simplification. Chapter 14. What the network tells us about life. Stand in a forest and look at the trees around you. What you see is the visible surface of something whose depth you cannot observe directly. The roots extend downward into darkness, branching into finer and finer threads until they become too thin to see without magnification. At the finest tips, where the root hair cells reach their maximum surface area, fungal hyphae are waiting. They enter the root space, exchanging molecules through cell membranes in a transaction that has been happening in forests for hundreds of millions of years. Those hyphae extend outward through the soil, threading past mineral grains and organic particles, finding the root systems of other trees and entering into the same exchange. The network they form is not a thing you can see or touch or point to. It has no edges, no center, no single component that is the network rather than part of the network. It is a process as much as a structure, a continuous flow of molecules between organisms whose separation at the genetic and evolutionary level is absolute, but whose functional life is intimately entangled. The network challenges some of the deepest assumptions built into Western biological science. The assumption of the individual as the primary unit of analysis. The assumption that evolution operates at the level of the organism competing against other organisms. The assumption that cooperation between species is always reducible to selfish interests that happen to align. None of these assumptions are simply wrong, but all of them are incomplete in ways that the mycorrhizal network makes visible. Life, it turns out, is more social than biology's individualist framework has always implied. Even organisms with no nervous system, no communication organs, no muscles, and no mobility exist in a web of relationships so dense and so functional that the boundary between them and their neighbors is, in many respects, arbitrary. A tree without its fungal partners is not a fully functional tree. A fungal community without photosynthesizing host trees cannot sustain itself. The forest without the network is a collection of doomed individuals. The network without the forest has nothing to network. This does not diminish the individual. The individual tree is real. Its genetic uniqueness is real. Its competitive interests are real. What changes is our understanding of the context in which that individual exists, which turns out to be, in the most literal and measurable sense, a context of physical connection to every other tree in the neighborhood. The question of what this means for how we understand life more broadly is not one that science can answer alone. It is a question that philosophy, ethics, and culture will need to work with as the science matures. But the science is already mature enough to say something definite. The forest is a network. The network is alive. And the life of the network is not the sum of the lives of the individual trees. It is something that emerges from their connection, something that did not exist before the Yasai. Connections formed and that ceases when they are broken. Chapter 15. The weight of what we don't know. It would be dishonest to conclude this account without acknowledging how much remains unknown. The mycorrhizal network is one of the most intensively studied biological systems of the last 3 decades, and the pace of research has accelerated dramatically as genomic tools, isotope tracing methods, and network analysis techniques have become available. More papers on mycorrhizal ecology were published between 2010 and 2020 than in all previous decades combined. The field is young, vigorous, and producing important results rapidly. And yet, we do not have a complete map of any single mycorrhizal network in any single forest. The species composition of fungal communities in most forests is incompletely characterized. The mechanisms by which chemical signals are transmitted through hyphal networks are not fully understood. The quantitative contribution of network-mediated resource transfer to individual tree carbon balance, as opposed to soil diffusion and other pathways, remains debated. The role of electrical signaling in intertree communication is plausible but not yet established. The evidence for kin-preferential resource transfer is intriguing but not yet definitive. The popular narrative about mycorrhizal networks has, in some respects, outrun the science. The concept of the Wood Wide Web, a term coined in a 1997 Nature News article about Simon's original study, has become a cultural touchstone, appearing in children's books, popular science writing, policy documents, and philosophical essays. It has been used to support arguments about forest management, about ecological ethics, about the nature of consciousness, and about the limitations of individualist worldviews. Not all of these uses are well grounded in the actual state of the science. A 2023 paper by a group of researchers including Justine Cast, Jason Hoeksema, and Melanie Jones, some of whom have been active contributors to the mycorrhizal network literature, argued explicitly that the popular version of the mycorrhizal network story has moved significantly beyond what the evidence supports. The paper, published in Nature, Ecology and Evolution, identified specific claims that have entered popular circulation but lack robust empirical support, including the magnitude of carbon transfer through networks, the existence and significance of mother trees as distinct hub entities, and the degree to which mycorrhizal network connections are preferentially beneficial rather than exploitative. This is exactly the kind of scientific self-correction that healthy fields require. The goal is not to dismiss the extraordinary biology of mycorrhizal networks, which is genuinely extraordinary, but to ensure that the extraordinary claims are matched by extraordinary evidence. Some of the claims currently circulating in popular culture are matched by that evidence. Some are not yet. What the corrective papers do not dispute is the core observation. Trees in forests are physically connected through fungal networks, and those connections facilitate the transfer of carbon, nutrients, water, and chemical signals in ways that influence the performance of the connected trees. This is established. This is real. This changes how we should think about forests. Everything else, the magnitude, the mechanism, the direction, the significance at the ecosystem level, is an active and productive area of scientific investigation. The story is not finished. It is barely past its opening chapter. Chapter 16. The forest beneath your feet. You do not need to be in British Columbia to encounter a mycorrhizal network. You do not need to be in a boreal forest or a temperate old growth to stand above one. You need only to be standing on soil where trees are growing, and that means almost any park, any urban greenbelt, any garden with a few mature trees, any patch of woods at the edge of any suburb anywhere in the Northern Hemisphere. The network is there. Below your feet, right now, if you are anywhere near a tree, an intricate web of fungal threads no wider than a bacterium is connecting root to root, transferring carbon from a tree that currently has surplus to one that currently has need, carrying chemical signals whose meaning we are only beginning to decode, and maintaining a structural memory of the forest's history that stretches back further than the oldest living individual tree above ground. This knowledge changes the walk through the forest in a way that most scientific discoveries do not. It changes not the landscape you can see, but the invisible one you are moving through. Every step you take in a forest is a step through a living network. Every tree you pass is a node. The root tips you are not seeing, a few centimeters below the leaves on the trail, are exchanging molecules with the root tips of trees you cannot see from where you stand. The forest is not a collection of trees. It is a community, in the most literal biological sense of that word. A community of interdependent organisms whose individuality is real, but whose existence is shaped at every level by their connections to each other. This does not make the forest safe or gentle or easy to understand. It is still a place of competition, of death, of parasitism, of organisms with no regard for each other, except what natural selection has inscribed in their biochemistry over hundreds of millions of years. The network is a product of evolution, not of cooperation as a value. But the outcome of that evolutionary history, operating at the scale of entire forest ecosystems, has produced something that functions in its aggregate effects as a system of mutual support. The trees are stronger together than they would be alone, not because they chose to be, because the network made it that way. There is something worth sitting with in that realization. The most sophisticated communication network on Earth is not the Internet. It is not the global telephone system or the satellite constellation or the fiber optic cables running under the oceans. It is the web of living threads running under every forest on every continent, operating without electricity, without coding, without any device more sophisticated than a cell membrane, and transmitting information that has been shaping the structure of terrestrial life since before the first animal crawled out of the sea. We did not build it. We have barely begun to understand it, and if we are not careful, we will destroy it before we finish learning what it knows. Outro. The closest living network to our sun is only a few centimeters deep. That phrase sounds like a trick, a linguistic joke, a way of making something small sound cosmic. But it isn't a trick, it is the truth. In the same century that we sent probes past every planet in the solar system, split the atom, sequenced the human genome, and photographed a black hole 55 million light-years away, we were still discovering what was happening directly below our feet. The signal that changed our understanding of forests did not come from a telescope. It came from a plot of British Columbian forest, where a researcher injected trees with radioactive carbon and waited to see where it went. It went everywhere. That was the discovery. We are surrounded by a network of living complexity that took hundreds of millions of years to build, that is more spatially extensive than any other biological system on Earth, that we have only partially mapped and only partially understood, and that we are disrupting at a rate our current models cannot fully predict. The terrifying part is not that trees can communicate. That is wonderful. The terrifying part is how long it took us to notice, and what we may already have destroyed before we did. If you are standing near trees right now, or if you will be soon, spend a moment acknowledging the network you cannot see. It existed before we did. It will, if we are careful, exist long after. And it is, in every meaningful sense, the foundation on which the visible forest rests. The trees are not alone. They never were. We were just too busy looking up to notice what was happening below.